Hello, welcome to Simplified Teaching. Now, today we'll be discussing photosynthesis in biology. The topics under photosynthesis, light reaction and dark reaction. Now, obviously, this light reaction and dark reaction in your younger classes, in your lower classes, you have studied this. But this year you'll be learning this in detail. So, what does this light reaction and dark reaction comprise of? Now, light reaction, okay, light reaction and dark reaction. Light reaction is of two types, okay, non-cyclic and cyclic, okay, non-cyclic and cyclic photophosphorylation. Now, what is this non-cyclic and cyclic photophosphorylation? That's what we'll be seeing, okay. But meanwhile, dark reaction, okay, well, that one, as we'll learn later, it will have three pathways: C3 pathway, C4 pathway and CAM, which we'll learn soon. So now, light reaction. What does this light reaction have? Okay. So, as I said before, non-cyclic photophosphorylation and cyclic. Now, what is this non-cyclic photophosphorylation? Is what we'll be seeing first. Okay, so non-cyclic photophosphorylation. You see, this non-cyclic photophosphorylation is the main pathway. Okay, it is the main pathway for photosynthesis. Okay, main pathway. Okay, it is the main pathway for all the activities that occur, and in this is non-cyclic photophosphorylation. Okay, where does it occur? It occurs in the thylakoid. Okay, it occurs in the thylakoid. Now, you'll be wondering what does it comprise of? Well, it's quite simple. Okay, well, as you've learned previously, photosystems. Okay, you know what are photosystems, right? Quantosomes plus PEA, primary electron acceptor. Two types are there: photosystem 1, photosystem 2. Okay, so now this non-cyclic photophosphorylation, it mainly depends on what happens from these photosystems. So now this one, it starts at photosystem 2. Okay, so from photosystem 2, what happens is, you see that primary electron acceptor is there, right? Okay, so now from photosystem 2, 2 electrons are given to primary electron acceptor. Okay, so now this primary electron acceptor will in turn give 2 electrons to PQ, CY, okay, which in turn gives it to PC. Now, what is this PQ, PY, PC? PQ, plastoquinone, CY, cytochrome complex, PC, plastocyanin. Okay, in each step, 2E, 2 electrons is given. Okay, so now what are these plastoquinone, cytochrome complex, plastocyanin? Well, you see, PEA is primary electron acceptor, but these are all other electron acceptors. Okay, they are also electron acceptors, but they are other electron acceptors. So now this is the step. In this step we get ATP. Okay, this is a very important step because this is the place. In this place we get ATP. Okay, so now let's move on. Now you see, obviously, now let's see what happens in photosystem 1. Okay, photosystem 1, what happens? Gives 2 electrons to primary electron acceptor, 2 electrons. Okay, which in turn gives 2 electrons to FT. What is this FD? Ferredoxin. What is ferredoxin? Yes, like previously, this too is an electron acceptor. Okay. This ferredoxin, now what is ferredoxin important for us? From this step, we get NADP plus. Okay, NADP reductase. Where is this NADP reductase present? It is in the outer membrane of thylakoid. This NADP is one of the most important compounds in photosynthesis along with ATP. Okay, so now you will be seeing, okay, here 2 electrons are given. Okay, obviously these 2 electrons, it's coming from these 2 electrons, it's going like this. Where does this 2 electrons come from? Nowhere. Obviously this 2 electron is coming from this photosystem 1 only, right? Okay, is it going back from NADP to photosystem 1? No. In the same way, in photosystem 2 as well, you see photosystem, it's all connected, right? So photosystem 2 to the same 2 electrons only. The same 2 electrons only goes here as well. Okay, whichever it's giving to that PQ, plastophenol, cytochrome complex, plastocyanin, the 2 electrons after producing ATP, that same 2 electron is only going through this process to give NADP. Okay, plus. But you see, these electrons that are lost, are these electrons coming back? No, these electrons are not coming back. They are permanently used up to provide NADP plus. Okay, so now since they are permanently used up to provide this NADP plus, what will photosystem 2 do then? Okay, what will photosystem 1 do? Okay, you see, because it's losing the electrons. Okay, obviously there can't be an absence of electrons now, can there? Okay, if there's an absence of electrons, then everything will go into turmoil. Okay, so because of that, a process called photolysis occurs. What is the process called? Photolysis. Where it occurs? Obviously from where the 2 electrons first comes, photosystem 2. Okay, this 2 electrons, it's going to photosystem 2. You see, this photolysis, it occurs in photosystem 2, and then from there electrons are supplied. Now, what is this process? Also called as, it's also called Hill's reaction, because the scientist who discovered was Robert Hill. Where does it take place? It takes place in the lumen of thylakoid. Okay, where does it take place? It takes place in the lumen of thylakoid. Okay, now what is the reaction for this? Okay, so this is what literally happens. 2H2O, okay, presence of Mn plus 2 plus, it gives 4 positive ions, 4 electrons, and oxygen. Now, this 4 positive ions you see here, this is the most important. What it does is, this 4 positive ions, right? It goes, and then what it does? It combines with N. It combines with NADP plus. Okay. Now, okay, this N. So it combines with NADP plus to give what? It combines with NADP plus to give NADPH2. From this NADPH2, we can again get more energy. Okay, from this NADPH2, it plays an important role in dark reaction, and it gives more energy. And for this 4 electrons, okay, that is being released. What is this 4 electrons doing? Okay, this is the most important part. This 4 electrons, out of this 4 electrons, you see, it is getting supplied to PS2. This 4 electrons is only getting supplied to PS2, and this in turn replenishes the electrons in PS2. Okay, this is why it's called non-cyclic photophosphorylation. If you study the full structure, this is what it is. It's quite simple. Okay, all you need to understand is photosystem 1, photosystem 2, 2 electrons are given. This 2 electrons, which is from photosystem 2, it goes on, okay, and final products it gives are ATP and NADP plus. But you see, these 2 electrons are completely lost. We can't get it back. So in order to replenish the 2 electrons that are lost, photolysis is happening. What is it called again? Photolysis. This photolysis, where it is taking place, okay, using what? In what? Photosystem 2. Okay, this is the reaction. I have written it down. 2H2O, okay, 4H plus, 4 electrons, oxygen. This oxygen is given to the atmosphere, which is, so it's not very important. The 4 positive ions that are being released combines with NADP plus, NADP reductase to give NADPH, and this 4 electrons is the one that is replenishing the lost electrons, maintaining the balance, everything in non-cyclic photophosphorylation. Now, let's move on to cyclic photophosphorylation. Cyclic photophosphorylation. What is this cyclic photophosphorylation? I told you that non-cyclic photophosphorylation is the main pathway. So obviously this is the additional pathway. What is this? This is the additional pathway. Okay. Now, where does this take place? I told you that non-cyclic photophosphorylation takes place where? Thylakoid. But this one takes place in the stromal lamellae. Where it takes place? Stromal lamellae. Okay. Now, non-cyclic photophosphorylation, it involves both photosystem 1 as well as photosystem 2. But this one directly it's involving only photosystem 1. Only photosystem 1. So now, you know what happens now in photosystem, that photosome is there, it is exciting the isolated chlorophyll A molecule, electron is getting excited, that excited electron, it's coming back to this photosystem 1. Okay, that excited electron is there. See, in the previous one, that excited electron is not coming back. Okay, excited electron in non-cyclic photophosphorylation, excited electron from photosystem 2 and photosystem 1, it's directly going. Okay, electron acceptors are accepting them and then they're giving the products. Okay, that is ATP and NADP plus. But in this, it is going a big cycle and it's coming back. Okay, well, not a big cycle, but yes, it goes in a cycle and comes back. So now what happens? Let's see. From photosystem 1, okay, as usual, it's going to primary electron acceptor. From primary electron acceptor, it's going to ferredoxin. Okay, from ferredoxin, it's going to plastoquinone. You know the sequence, right? It goes to plastoquinone, then it goes to cytochrome complex, then from cytochrome complex to plastocyanin, and then finally it comes back to photosystem 1. Okay, it goes. So now in this, okay, in this you should know that there is no photolysis. Okay, no photolysis, no ATP, and no NADPH2. Okay, none of this is produced. Okay, none of this is produced. With this dark reaction, with this light reaction is over. Now we will move on to dark reaction. Okay, dark reaction. Okay, this dark reaction, just because it's dark reaction doesn't mean it happens only at night. Dark reaction, it can happen at any time of the day. Okay, now what is the importance of this dark reaction? Nothing but in this dark reaction, ATP, okay, NADPH. Now when it is produced, right? When is it produced? Okay, in non-cyclic photophosphorylation, we are getting all of this. Okay, now this thing that we're getting, right, these two compounds, it is used to reduce carbon dioxide to glucose. It is used to reduce carbon dioxide to glucose. By reducing carbon dioxide to glucose, process of photosynthesis gets over. Now, there are three different pathways in dark reaction: C3 cycle, C4 cycle, CAM. C3 cycle, C4 cycle, and CAM. This we shall discuss in the next class. I hope everything that I did today was clear. Please post any doubts in the comment section. Please like, share, and subscribe. Stay tuned for more videos. Thanks for listening.